Hi, this is Yanhong Liu. Today I will talk about an experiment to determine the effects of novel phytase on growth performance and metacarpal bone ash in William pigs. First, I will briefly introduce the phytate and phytase and also show the objective of this experiment. And then I will show the details about how we conduct this experiment, and then move to the results, and finally I will give an overall conclusion. Why phosphorus is important? Phosphorus is a key structural mineral in bones and teeth, and phosphorus is a component of DNA and RNA. Phosphorus is critical in the structural integrity of our cellular membranes, and it also is very critical in metabolic energy systems. Therefore, a consistent supply of phosphorus is required to support pig production. There are two types of feed ingredients to supply phosphorus in animal feed. The animal origin feed ingredients do not contain any phytate, but a high percentage of phosphorus is bound to phytate in plant origin feed ingredients. Inorganic phosphorus source also available in feed market, and these inorganic sources do not contain any phytate. There are two common phosphorus sources used in pig diet, which include monocalcium phosphate and dicalcium phosphate. So what is phytate? The full name of phytate is myo-inositol hexacephosphate. Phytate has anti-nutritional effects because phytate can form a complex with calcium, phosphorus, protein, and trace minerals. Because of this complex, phytate can reduce the digestibility of these nutrients. So therefore, they can increase the environmental pollution, such as they can increase the nitrogen excretion and phosphorus excretion. Because of reduced digestibility of nutrients by phytate, there are decreased profitabilities. Phytase is an enzyme to catalyze the hydrolysis of phytate to inositol and inorganic phosphorus. And after the hydrolysis, this inorganic phosphorus can be utilized by animals. There are two different types of phytase in the market: the fungal phytase from Aspergillus or Panthera species, and the bacterial phytase normally from E. coli. So adding phytase in animal diet can reduce the inclusion of inorganic phosphorus and calcium source in animal feeds because they can increase the digestibility of calcium and phosphorus. Adding phytase to animal diet also has beneficial effects on the environment because they can reduce the phosphorus and nitrogen excretion. The objective of this experiment was to determine the effects of a novel next-generation phytase on growth performance and metacarpal bone ash of William pigs. The brand name of this phytase is Sebenza Phytoverse G10. A total of 160 pigs with 35 days of age and initial body weight around 9.79 kg were used in this experiment. The experiment was conducted with 2 phases and 2 weeks for each phase. There were 4 diets in each phase based on corn and soybean meal. The first diet was a positive control diet with a digestible phosphorus metal requirement. The second diet was negative control diet with 56 to 58% digestible phosphorus compared with positive control diet. The third and fourth diet was negative control diet plus 250 or 500 FTU per kilogram Sebenza Phytoverse G10. There were 10 replicate pens per treatment and 2 barrels and 2 gills per pen. This table shows the negative control and positive control diet in phase 1. From this table, we can say both negative control diet and positive control diet contain similar amount of corn and soybean meal. The only difference between negative control and positive control diet was the negative control diet contained 0.23% monocalcium phosphate, but the positive control diet contained 1.03% monocalcium phosphate. This slide shows the nutritional composition for these 2 diets. From this table, we can say negative control diet and positive control diet contain similar amount dry matter and crude protein, but they have different amount of ash, calcium, and phosphorus. So negative control diet contain 4.07% ash, 0.75% calcium, and 0.41% phosphorus. But the positive control diet contain 4.49% ash, 0.82% calcium, and 0.56% phosphorus. The growth performance was checked by calculated average daily gain, average daily feed intake, and gain to feed ratio. At the end of the experiment, 2 pigs per pen, 1 barrel and 1 gills were euthanized to collect 3rd and 4th metacarpal bones from the right foot. The bone ash and phosphorus were measured in these bones. Our data were analyzed by ProgGLM with completely randomized design. The statistical model include diet as fixed effect and pen as experimental unit. The orthogonal contrasts were conducted to test linear and quadratic effects or a graded level of phytase. Move to the results. The first figure showed the overall average daily gain. So first, let me set up the slide. The y-axis, we have the average daily gain, and x-axis, we have 4 dietary treatments. The green bar represent positive control diet, and light blue represent negative control diet, and light yellow represent 250 FTU per kilogram phytase, and orange bar represent 500 FTU per kilogram phytase. So from this figure, we can say, compared with the positive control diet, pigs fed negative control diet had lower overall average daily gain, and adding phytase to negative control diet linearly increased overall average daily gain. For the overall average daily feed intake, again compared with the positive control, pigs fed negative control diet had lower average daily feed intake, and adding phytase linearly increased average daily feed intake. Overall gain to feed ratio, compared with the positive control diet, negative control diet reduced the gain to feed ratio, adding phytase to negative control diet linearly increased the gain to feed ratio of William pigs. This slide shows the bone weight. This is a fat-free bone weight. So again, compared with the positive control diet, pigs fed negative control diet had a lower fat-free bone weight. And adding phytase to negative control diet linearly increased the fat-free bone weight. And this slide shows the bone ash weight. Again, compared with positive control diet, pigs fed negative control diet had lower bone ash weight. And adding phytase linearly increased the bone ash weight. For the bone phosphorus weight, compared with the positive control diet, pigs fed negative control diet reduced the bone phosphorus weight, and adding phytase to negative control diet linearly increased the bone phosphorus weight. We calculated the bone ash percentage from bone ash weight divided by the fat-free bone weight. So this figure shows the percentage. Compared with the positive control, pigs fed negative control diet had lower bone ash percentage, and adding phytase linearly increased the bone ash percentage. For bone phosphorus percentage, again compared with the positive control diet, pigs fed negative control diet reduced the bone phosphorus percentage, but adding phytase did not affect bone phosphorus percentage. Based on the results shown above, the overall conclusion for this experiment was this novel phytase supplementation to a phosphorus-deficient diet based on corn and soybean meal improved phosphorus utilization by William pigs. Thank you so much for listening to this presentation. If you have any question related to this experiment, please go to our website. You can find the research summary there, and also you can contact Dr. Stan for more information.